There we go. Now we're live. Hey guys.
All right, we'll get Instagram going too.
Awesome.
All right, we are good.
Sweet. What's up, guys? Soda balls, sweet slithering delights. Good to see you guys again. BJ the Crow and Adrian. What's up, guys?
Thanks for joining in, and we are going to have Tommy joining us. She said she's running around. Oh, it looks like she's coming in right now.
Oh,
Told
awesome.
her to say if you got home quickly.
Hey, Tommy.
Hello, how are you guys?
Doing good.
Good. Glad you made it.
Yeah, I literally made a sandwich and went upstairs to do something else, and Waffles licked it while I was upstairs, so I had to remake it. So that's why.
No.
Dang it.
Dang cat.
Sure. Oh my gosh.
That's the one good thing about, well, okay, there are multiple good things about Potato, but like this little jerk over here in the chair behind me, she's adorable, but she's not food motivated, which is nice because she will get into everything else.
Yeah. Yeah. I don't have any cats anymore.
I missed you guys.
I had 4 for a long time, but
wait, so who was Waffles? Your dog?
Waffles is my dog. One of my dogs.
Oh, gotcha.
I have 3 dogs now. I used to have 4.
Oh.
Looks like people are rolling in. Yeah, for sure. On top of everything else.
Yeah. I'm glad things went well last week. I was moving into the new place. You can still see boxes and empty space, but I'm here and oh my God, I love it.
Oh, good. Good. Moving is hard.
Movers though? Oh my God, it's the first time in my life I've ever hired movers.
Was it worth it?
Oh my God, yes. Oh my God, so worth it. Like I've usually done the either do it myself or invite some friends and lure them with pizza and beer.
The pizza party.
Oh yeah.
Exactly. I know that life for sure.
But all my friends are getting bad backs and bad knees and sciatica. So
what? There's no way. You're like, how old are you?
I'm 29. I'm 29. They're just they should
way early.
My friends are old.
They're old young.
Exactly.
Yeah. We do have quite a few rolling in on Instagram too.
Nice.
Orly 420, Hogalicious, Snake Morse 1, Killer Cold Bloods, Sil Royal, Down the Hole Exotics, I-11, Randy Parks, Killer Bloods, Central Valley Morphs, Royal Pieds. Yeah, Ball Pythons Project. There's still more rolling in. Twisted
That's
Python.
awesome.
Yeah.
Sciatica started at 19. Oh my God, I can't imagine. That's how one of my friends are. I'm so sorry.
That is very young. That's no fun at all.
That's extremely young, yeah.
Yeah.
Updates. Paratrooper Pythons is looking for updates.
Yes. So, and then we've got rodents and royals coming in late. You're not very late.
No.
So one of
you're right behind me.
Yeah, one of the updates was what our video went up for on Tuesday.
Yeah.
Rodents and royals. It was an animal from them that came in that was clearly a blue-eyed Lucy, but for our blue-eyed Lucy panel, only tested het for phantom. And then we happened to have one other customer that had sent one in, and it only tested het Mojave, but it was clearly a blue-eyed Lucy. And so they kind of kicked off a whole bunch of work that I did behind the scenes, trying to figure out what was going on. And we found another mutation in the blue-eyed Lucy gene, the same gene that Mojave and Lesser and, Phantom and Mystic, all those things, that it's in that same gene, but it's a different location. It's a different mutation that we ever knew about before. And so what we're trying to figure out is this a brand new mutation that we didn't know about? Or after looking at the history, what we saw was back in 2001, the first butters were produced by the Bells and the first Lessers were produced by Ralph Davis. And then 2 years later, Ralph started producing from a male he called his Butterball. And it was a male he got the same year the Bells got their butter. And so they kind of assumed it was probably the same, but don't really know if it came from the same location that Ralph got his original Platy Daddy from that made the lessers. It could have also been lesser, but he might have been selling those as butters that whole time, assuming since he got it the same year as them and didn't know about the lesser at first because he got that Platy Daddy. And until he hatched out babies and worked on it a few years, then he realized, oh, there's actually 2 mutations in this Platy Daddy. And any of you that have been around for a long time know how complicated that was to figure out how that worked. But anyway, the bottom line is we are asking for people that have had lessers and butters like back into like the mid 2000s or earlier. If you got them directly from the Bells or from Ralph Davis, that's even better. But we want to get more sheds in and see if what we found is actually a separate mutation that the butter actually is. It's just butter, or maybe the Bells had the butter, but they had another mutation that is also in the blue-eyed Lucy complex. So that's one big update. Those of you that didn't see that episode yet, we had Bob from Bob's Balls because he sold the animal to Rodents and Royals that produced that all white one. Anyway, it was nice having Bob on, and we also talked a little bit about the snake he's got in that picture there. That's a cool story too. What was it we were saying? It was some kind of cross. You had a name for it, Kayla?
Yeah, it's a cottonhead, or he strongly suspects that it's a cottonhead. I can pull up like the actual photo, but
just based off of size or because of the like the way the head looks.
Size, yes, size, coloration, behavior, and geographic location. Like cottonhead, like basically copperhead cottonmouth crosses. They have been found in the wild, and this is an area where they both are. And this was just like a big old possibly gravid female who, like, normally when you find a copperhead, they're pretty chill and they're just like, oh, I'm going to get away, or they just freeze up. Whereas this one was kind of like posting up and kind of like opening its mouth a little bit like a copperhead or a cottonmouth.
Okay, or a cottonmouth. So it was doing cottonmouth type behaviors.
Yeah.
Oh, that's crazy.
Right?
That's crazy.
Yeah, no, it's cool. And he got some nice pictures of it.
Yeah, I watched the episode, but I missed that part. It might have been at the beginning or at the end or something. I was as per usual doing 4 things at the same time. So
mood. Yeah.
It happens.
All good.
That's really cool.
Yeah. Sweet Slithering Delights is asking if we've seen anything weird like that with leopard tests. A few others to talk to had thought that you had Leos, but it tested negative.
Yeah, I mean, when we run thousands and thousands, tens of thousands of samples, we definitely see weird stuff pop up. In leopard, it's fairly rare, but we have had a couple where people felt like, okay, I really think this is a leopard, but it's negative, or this doesn't seem like it's a leopard at all, but it's positive. You know, weird stuff. Like we said, we guarantee it at 99% that we, do the validation before. But if we do, 100 or a couple hundred and validate it and feel comfortable with it, but then we have 1,000 come in, we might learn, oh, okay, there is something that we didn't see in that first 1 to 200. So I would say leopard is very rare. We see anything weird. But the really helpful thing to us is when that happens, definitely message us, let us know. And if we can kind of trace back and see, okay, how about the sire to this or the dam, whichever one was a leopard, let's test that. And if it's positive for our leopard test, but this animal's negative, then that's even more evidence that it's actually really negative. But if that adult that's negative, that looks like a leopard, is negative, then we can kind of trace around and see, okay, maybe this is another mutation that looks like a leopard, but it's not. Or if we find enough evidence, it could be that our test isn't picking up all leopards, like what happened with Ultramel and albino and several of them now, where we find out there is a secondary mutation. So I haven't seen it very much with leopard, but the more we trace lineages, like when Kayla puts up the thing asking for people to send sheds in, that's why we ask for any lineage information, because if something weird pops up, then we can kind of chase it a little bit. So
yeah, here, let me hunt it down real quick. Where's my thing? There's my thing. There we go. I lost my thing. There we go. Right there at the bottom right. Lineage info.
Yes. So that helps in times like that. So it doesn't matter what you're testing. Like back when COVID was happening, you heard, what's the false positive rate or false negative rate, and what's the CT value? Like there's, things when you say in general, does this test work? Then yes. And if you can say it's over 99% correct, that's great. But if it's over 99% correct, and then you run 10,000 tests, that means you're going to have, several that are not correct. It doesn't change the fact the vast majority are correct. And then we just have to drill in and see, okay, is it a problem with the test? Is it that there's another line that we will have to develop a test for? We just have to figure that out. And it takes some time to do that. And there's no way to get around that. We just have to start running a test and figure out if we see problems, just keep improving it. And so you all that pay for the tests, when something weird comes up, let us know and work with us. Then we can make that test even better. And many of you have already done that. We're very thankful for that. And our tests are even better because you all are interacting and helping us do that.
Oh, yeah. Prophecy Exotics. I must confess, I send full sheds instead of quarter-size pieces. As long as they're super, super dry and they aren't going to like mold in transit, it's good.
Yeah. And you're also paying more for shipping. So.
Yeah, that too.
It's up to you if you want to do that. But I think I've had people send in 4 or 5 samples in just a normal envelope with one stamp. So if you're sending small pieces in smaller Ziplocs, you can do it really, really cheap. So that's the other part. If you send full sheds, you're going to have to pay more for the shipping.
Yeah. All right. We got people calling out their
shed test that they want.
Yeah. The shed test they want. So redheads. Oh yeah.
Get people to
tri-stripe
head sheds. Yeah.
tri-stripe.
Tri-stripe. Yep.
Yep.
Tommy's helped. There's multiple people that have sent in decent numbers, like more than 10. And I think Tommy was part of hunting those people down.
Yeah, I've definitely tried really hard. The tri-stripe community is kind of like divided right now. There's some people who are like, we don't want the shed test because they want to be able to sell their 66% hets. So we're like battling right now. Like, and I'm like, but I want to know which ones are minor hets so I can sell the ones that are not.
Yeah.
So
I believe the other testing companies, I think Charlie and the other people that are, developing tests, I think they're working on it too. So hopefully one way or the other,
it
gets, yeah, absolutely.
Yeah. I'm going to put this wiggly guy away real quick.
Yeah, wiggly guy.
Oh yeah. Well, I'll show him off real quick. Mr. Lemon Bars. The man,
I've seen him before.
The legend. Oh yes, he is.
Is he like your favorite boy?
Jay says that he's my favorite boy. I say that whatever one is currently in my hand is my favorite. But as like, how you like sing songs about your pets, you just make up little songs.
No, I've never done that.
What? Really?
No, I'm kidding.
Just like what? But whenever it happens, it's either about the cat or this guy. So, I mean, strong possibility.
Patricia says she can finally send a research shed because she just got a visual try like a couple weeks ago.
Yep. There it is. Right on.
Yep.
Yeah. We appreciate it.
Yeah.
And 666 Serpent said, I'm going to get my monitor shed to you at Daytona.
Yay! Awesome. Monitor sheds.
Yes. Known sex monitors, Heloderma, blue tongue skinks. All that would be helpful.
For sure.
Yes.
Oh yeah. How much material is needed for testing scale clippings? I'll pull up that video that we took, Ben.
Yeah, do that. So really to answer what you're saying specifically, and the video is less than a minute, so we can play that. But it really looks like an eyelash. It's just the shaving off of the edge of a ventral scale. The part of the ventral scale, the very outer edge that's clear, you can just like the same scissors you use to pip eggs. If you do pip or cut eggs, you just shave off the edge of that ventral or belly scale. And it literally looks like an eyelash, and it works great. You can see there.
There it is. Kind of.
very
There
small
it
piece.
is.
It does go into
Yeah.
focus at some point.
Here, I will.
But that's how small it is. It's hard to go into focus.
Yeah,
Oh,
yeah.
wait, I can probably improve the quality. Yeah.
And if you have
1080.
Oh,
if you have a
there you
second person helping you here, I'm just doing it, one person. But if you have a second person kind of helping you hold, and one person hold, one person cut, it's a little easier. But you just kind of shave off. See how I'm kind of holding up and just getting the clear outer part. It's very, very small.
It focuses in a second. I was taking this on my phone.
And
There we go.
then when you
Ta-da.
put it in the bag, just kind of seal it good so there's no air, and then just kind of circle it. And that way we'll know where to start looking.
And send
in
like
this general area.
send like 3 of those, and you're golden.
Yeah. Send at least 2 or 3, because if we have a problem, then we want to go to the second one. Sometimes a run, goes bad on us. It has nothing to do with any of the samples, just something with the machine or with some of the reagents or whatever. So every once in a while we'll have to go back. And then if you're doing multiple animals, just go ahead and use some rubbing alcohol from the store and flame it, just to get rid of any DNA that may have been left behind. And then you can go to the next snake.
Yep. Yeah, just a belly scoot. You don't have to go for anything else.
And just barely a little bit does it. We don't need a lot of material. Just a little bit is plenty. And I was telling somebody, I think there was a video that someone did. They worked with Charlie on this one, but and it's something done in birds as well. But if you have an animal you're really excited to get tested, whether it's for sex or for morph tests, when it's in the egg, you can actually even get just a little bit of the blood. So if you just took a toothpick, you just get a little bit of the blood in that egg, and then put that toothpick into an open Ziploc and let it dry out in that Ziploc. Then once it's dry, seal it. You can send that, and we'll be able to get DNA from that too. But then you just have to make sure, like if you're sending a whole clutch like that, you might have to separate the eggs before they hatch, or somehow make sure you know which toothpicks go with which ones. But if it's just like one from the clutch you're really excited for, you're for sure not going to forget what toothpick that goes to or whatever. Then you could do it that way too.
Yeah. And when sending clippings, I usually do 1 per test plus 1 extra. Yeah, that's smart. Yeah. I've done it on tiny colubrids too. Not like hatchling size, but like when I got some, I think yearlings, like very small yearlings. It was a pain, but I did it.
Yeah, that was impressive.
On colubrids?
Mm-hmm. Baby rat snakes. Yeah,
that's really rough. Yeah.
I had my boyfriend holding poor little wiggly thing, and I was just like, just barely. I was using cuticle clippers actually instead of like shears, but it worked.
Yeah.
Nice. Wouldn't do it again, but no, it worked.
No. I wouldn't even want to do it on a baby piebald, but I'm old and so are my eyes. So there's that.
Oh. Yeah.
So yeah, the question I popped up.
Oh yeah.
How would a champagne look pied if not pied or het pied? We don't really know for sure why, but that's how champagne reacts. If it's champagne and it's a het pied, a lot of the time it'll look like a full-on pied, and sometimes even not. Like we have, and like Tommy says here, if we have cinnamon or black pastel along with champagne, and no pied at all, it can look very much like a pied.
It can throw a really hellacious ringer for sure, like a really big ringer that's like, so it looks like a low white pied.
Yeah, all of our Cisco stuff looks like it could be pieds, but none of them, they don't have pied in them at all. They're just hypo. And then they have Orange Dream. That's one that Tommy got.
Black pastel. Yeah.
Yep.
Black pastel. Yep. Yeah. The 8-ball complex does definitely tend to throw those ringers in champagne combos.
That's so cool.
Yeah.
I like champagne a lot. I feel like it's a very underworked thing, but I can understand why in some ways, but I haven't had any of those problems. So
yeah, I think with clown being so popular, and it's difficult to have champagne do something interesting with clown. That's one reason why a lot of people don't like it. But if you're not doing any clown stuff, like the hypo stuff we've done with champagne and different mixes is awesome. You get great color, and a lot of the time it'll make it patternless with patches of white, patches of orange, or like kind of purplish if you don't have the black pastel in there. Like some of ours, we have enchi instead of black pastel, and it'll be bright orange and stuff like that. And so anyway, it's pretty fun.
Yeah, I really dislike the fact that every incomplete dominant has to do well and clown to be popular. that makes me crazy because I don't work really in clown, so being the super is lethal is restricted pairings a lot. I don't know how it would restrict pairings unless you have a very small collection. Because I mean, you don't have or if you're trying to do like triple recessives or something with the same girl back to back to back. But I mean, you shouldn't have a problem. Just avoid pairing them together.
Yeah.
I didn't know the super champagne was lethal.
Yeah, just like super spider.
Gotcha.
It gets hairy in the whole spider complex. There's a lot of the like the super cypress can be wonky. Cypress and like Woma can be wonky. There's definitely some strange things.
And Woma spider can be bad.
Yeah.
Oh no. Sure. Man, it's so strange.
Yeah, anything in the spider complex with spider other than blackhead, avoid like crazy, pretty much.
Yeah.
So.
Yeah, and I think some people have had super spiders live for a little bit too, but not for very long. Usually. The same complex seem to have strong. Yeah, so they're in most cases they're on different chromosomes. There's several things there: leopard yellow belly, and 8-ball. I'm pretty sure are on different chromosomes, but there's definitely like in dogs the color with labs. We've talked about this a few times, but like a black lab versus a chocolate lab versus a black lab, it's actually two different genes on completely different chromosomes interacting with each other to give you those 3 different possibilities. So you can definitely have mutations on completely different chromosomes, completely different genes, but they can play a part in the same pathway, and they can affect how something looks or whether it's viable. The other thing is Justin Kabelka's clutch, where he bred a cryptic to a puzzle, and all the babies look like cryptics. So puzzle,
oh yeah, I remember that.
Cryptics seem to interact, and then also we get weird results whether something looks like a clown and a puzzle or doesn't, and it can test out to be homozygous for both. It seems like there's some interaction, even though we know clown and cryptic are in the same gene right by each other, but we know puzzle's on a different chromosome. But there seems to be some interaction. They seem to be part of the same pathway or something like that. So it definitely can happen.
Yeah. Oh, slight direction change, but just because we were talking about butter earlier, this thing that you were sent just before we keep going on.
Yeah. So Jeff Byers, I think he works with Will Banks Reptiles. But thanks for sending this in, Jeff. Yeah, it's pretty interesting. Saying that Earl Jones, I think he's in Texas, if I remember right, breeder in Texas, said the difference between lesser and butter. When you breed them to leopard, a lesser leopard will stripe out, whereas a butter leopard will just get more of a wacky pattern. So that's kind of interesting. That's something I would put out to you all watching this, if you've seen something similar, or have any experience breeding leopards to lessers or butters, and what you see from that. That'd be kind of interesting. Now we could take ones that people breed and test them and see if it's actually that lesser mutation we've known about for a couple of years now, or if it's this new one that maybe is the original butter, but we're not sure yet.
Yeah. I love this. All the butter believers being vindicated after all these years must be such a good feeling. Yeah.
It's kind of looking that way. The other thing that's possible is butter and lesser are the same, but the Bells not only had butter, but they had another mutation too. But I would say the more data that's coming in, that seems less likely. I think it's more likely that we found the original butter, but we don't have enough data to know for sure yet. So.
Mm-hmm.
So yeah, please keep spreading the word. I've heard people say they've had people call and ask about, lesser versus butter. So it seems like people are getting the word around. So I like hearing that.
Mm-hmm. Geeked was mentioning hypostatic mutations when we were talking about the 8-ball complex before. Going back to that. So hypostatic versus epistatic. I don't know those words. Ben, do you know those words?
I know epistatic. Hypostatic, I'm not familiar with.
Yeah. Google time.
Epistatic.
Google time.
That just means epi means above, so it's something outside of that general area.
Hypostatic gene is one whose phenotype is altered by the expression of allele at a separate locus. Okay, so kind of like the so like the lab situation you were describing.
Yeah, that's when he said that was during the lab conversation.
Makes sense. Gotcha.
I hadn't heard that term. That's cool. But that's essentially what I thought epistatic meant. But maybe there's a difference when it turns it down. It's a hypostatic. I don't know.
Let's see.
Patricia. Patricia's asking about homozygous acids.
Ooh.
I don't think we've tested one to be homozygous yet that I can remember.
She says you had a confusion. Oh, it was produced the gene on both sides. So she's saying the pairing, not the test. The pairing had confusion on both sides of the animal.
Acid confusion, wondering if any animals tested homozygous. I was unclear on. Yeah, I don't think we've had one test homozygous. I have a confusion produced with a gene on both sides. She's saying confusion on both sides.
Yeah, both the sire and the dam had confusion on both sides.
Yeah,
so
hopefully we'll have a confusion test soon, and we'd be able to figure that out.
Yeah. Is there a test for acid? Oh.
Yeah, there's been a test for acid for a while.
Yeah.
Just no confusion yet.
I don't know who would have tested static against confusion. I don't know that anyone has a confusion test, but and we just don't have very many static sheds. I think I've only gotten 1 or 2 in, so I just don't really have enough to, even if I ran those and they said the same thing, it's still not really sure if the person knew for sure what they had or whatever. So kind of need like 5 or 10 statics from 2 or 3 different people. Like if 2 or 3 different people sent us 2 or 3 each, then we can be a little more sure about it. But there just doesn't seem to be that many statics out there. If you know someone with them, have them send them in.
Yes, please.
I think that Devin had said Fred Kick has a bunch of them, or maybe I'm remembering it wrong. I talk to too many people every day. My brain can't keep it all straight. It's tested against
acid. Okay, he had it. Yeah. Okay.
Acid, yeah.
All good.
Yeah, I don't remember anything testing positive for acid except for acids. I think there's only one time so far we had one come in labeled as a confusion, and the mediocre confusion actually had a couple tests that were like 70 to 80% correct on confusion. Both of them were negative for confusion, but it was positive for acid. So that's the only time one was different than what somebody sent it in as. So it was likely actually an acid and not a confusion. But yeah, I don't remember any statics testing positive for acid. But like I said, I think we've only had a couple come in. So
yeah. Oh, okay. Yeah. Then Fred Kick would have some.
Yeah. So if anyone knows, tell them to send sheds in.
Yes, please.
Yeah. Let's go, Fred.
Yes.
Everybody tag Fred. Respectfully.
Respectfully tag.
Respectfully.
That's funny.
Yeah.
So when we were talking about champagne, we had a couple comments in Instagram. Saltaire and serpentines. So Andy from last week, hey, He said, "I heard it can have a wobble like spider, but never seen a wobbling champagne." I don't, I don't really think I have either. Not if it's just
I
champagne.
I have one.
You got a wobbler, so every once in a while.
Yeah. I
Okay.
got a wobbly baby, yeah. But that was the one out of all of them. So
Yeah.
it's not like all of them by any closer on the monsoon stuff.
Fair enough.
And down the hole exotic said champagne wobble is the only reason I'd avoid it, but I've also never had a wobbling champagne. And then Sinful Doll 6969 shed a girl who was supposed to be GHI cinnamon champagne. She was negative for everything but champagne, but looks pied. So yeah, she's probably champagne and
Yeah.
something else that's making it look like a pied. But yeah, we're still the 8-ball complex. It's kind of a messy part of the genome, and it's been difficult to find those and get tests that work well. So maybe it's something else in the 8-ball complex that we don't have yet. So we'll see. Monsoon stuff. I am hopeful. Don't quote me on it, but I'm hopeful in the next week or two we'll have an episode where we'll talk about a monsoon update finally. But not today. Don't have anything for sure today. But I think in the next week or two we have a good chance that we'll be able to have a good talk about that finally.
This time for real.
We'll see.
That's what I tell myself. That's what I tell myself every time. So. Eventually, it'll be right. Maybe.
Yeah.
Right into Royals, I actually have a champagne het pie that does not look pied. It's just solid colored.
Ooh.
Somebody, because he was talking about, or they were talking, I don't know if it's a he, was saying that it's a poor man's pie.
Yeah. So I guess it can happen where they don't look like a pied.
Yeah. Tommy has everything.
So Brian, on this one, if you message Sean and ask Sean that, Sean will know the most up-to-date information and we'll be able to tell you. I don't help with any of the bulk stuff, so I can't tell you for sure what is and isn't cool, but Sean knows what the latest is on monsoon, and so he'll be able to tell you what will be good, and same with any of the rest of you that are sending in multiple sheds fairly frequently. Just ask Sean what we can and can't do right now for monsoon. And then, like I said, within the next week or two, there's a good chance. It's broken my heart so many times that I'm not getting my hopes up too much, but I do think in the next week or two, we will have a very comprehensive update. So everybody will know exactly. And like we've said before, we're all working on this. Charlie here in the US, Heather up in Canada, Jurgen in Europe. We're all working on it together, sharing information. It's been a big pain in the butt. We've all said that purposely not adding up the money we've spent on that because it would be so depressing. It's been a lot of money and a lot of time, but yeah, in the next week or two, I think we'll have a really good comprehensive update that will let people know where we're at.
Awesome. The sun does not set on the mission for monsoon.
It's that way.
Any update on the stranger tests?
Not yet, but I will. I do have some data I'm starting to analyze now, and some of that is stranger stuff. But I'm waiting for, we're also updating our tools, and I'm still waiting for that update because that will help me exponentially in being able to pinpoint which hits are the most likely to be correct. So we spend less time designing and testing and validating to then have it turn out to not be right. So this new tool should help me increase the probability of hits that I chase being the correct ones. So I'm still waiting a little bit. I'm still hopeful that that's coming in the next week or so, but I've been hoping that for a little bit, but I know the person working on that tool has been at it for like over a month, pushing hard. I know we're doing all that we can, and as soon as we've got it, Stranger will be one of the ones. Stranger, Paint, there's quite a few. Suma, so the mahogany, there's several of them that we'll all focus on with this new tool, and hopefully we'll pop many, if not all of them, loose.
Awesome. Hi, Stephen and Courtney.
Hey.
Let's start a couple of these. Oh, I already answered those. Awesome. Totally unrelated, but look at this little orange guy. He's a rough green snake. Isn't he cute? He was wild caught, and I've been following him for like a year because usually when I see that and it's like a baby, I'm like, it's probably gonna die. Like the world of diminutive colubrids is so brutal, but he's been surviving and eating, and it's been like a year.
Wow.
Yeah.
I'm gonna send you a picture, Kayla, of this animal.
Oh, yeah?
Hold on a second. Yeah. I forgot I meant to send it to you for the show, but it's it was a wild caught Western Diamondback that is a mutation now, and it's insane looking. So I'll send that to you.
Awesome.
Cool.
Yeah, I'd love to see it.
Like that.
Yeah, it's really interesting. I sent them a message to ask about it because I was like trying to find out more information. They're still pretty cryptic about it. So
Ben and I fighting with the icons.
What are you doing? You're clicking things.
If we both click like a comment or like to hide something at the same time, then we'd never know what it's gonna do.
Exactly.
Yeah.
Dragon Ruler 4, the petition to call it the mean soon, since it seems to be bullying all the shed testers.
It is.
I'll put my vote in for that. It's been
it truly is.
Ooh.
Yeah. I sent it. Yeah, definitely. We have that happen all the time on Morph Map. Like I'll be clicking on something because I'll see something and I want to make sure that I get to it, and then he'll click it at the same time or click it off, and I try not to do it anymore.
Yeah.
I can't look at chat while I'm talking because I'll go crazy.
Oh, I'm the same way.
This hypothetical, this is something that Randy, I think is it Snake Genetics one? Or I can't remember what he goes by, but Randy brought up, the possibility of bringing morphs from one species to another. It's certainly possible. I haven't seen any evidence of it, but we also haven't looked, super close. It would surprise me if that's happened with ball pythons. I think it'd be more likely that something popped up in ball pythons and they took it to another species rather than from another species into ball pythons. But we have sequenced enough whole genomes for ball pythons that if there was a suspicion of a specific morph coming in that way from Burmese or from Retic or whatever, we should be able to have pretty clear data, whether that is a possibility or whether it's obviously a pure ball python. It probably wouldn't really work for us to like just test randomly, but if you said, okay, this specific project, I think this might have came from bloods or came from retics, but it takes time and money to do that, and unless somebody has enough suspicion to make it worth going down that path, then we probably won't.
Yeah, I remember Randy bringing that up last time.
Oh yeah, GHI spider burn ball. I think I saw that picture. Like a incomplete dominant or dominant traits, they'll just like pair it from a ball python to a berm and then morph burn
Some
balls.
of them pull over there. Yeah.
Yep.
So they said that one went up today on Instagram. I'll have to look for that. That's cool. It's messy. I haven't done any of those crosses, but I definitely thought about it early on about crossbreeding to other pythons to get pied into them, because pied was by far the coolest one in my mind. But now it seems naturally that it's come up in other, what we see in bloods. It's definitely not from ball pythons to bloods. The blood one is its own mutation. But we haven't looked deeply in retics or berms yet at their pieds. But yeah, I think it's kind of a fun and interesting. Hey, Charlie.
Hi, Charlie.
Kind of a fun and interesting question. But I just, for me personally, I didn't want to make the byproducts, and I didn't really want to cull them all. So I didn't ever go. But people that feel like that's worth it, and if you have a pet that's a berm ball pet, and you like it, you dig it, nothing wrong with that. I didn't want to go down that route myself, but it's definitely interesting to think about.
Spider burn balls also have wobble. I don't know.
No clue.
Yeah, I don't know either.
I don't do any hybrids. I don't do any hybrid stuff. I like the look of it, but I don't do any of it.
So y'all can find that on Instagram and ask somebody.
Yeah, exactly.
Yeah. Did I get the pictures you sent of the turtle? Yes, I think we showed those. Was it like 2 weeks ago? I'm pretty sure. But yes, we did get those, and I loved it. I might have to throw those up again because they're. I'd have to dig through my email. But yeah, if anybody else wants to send stuff like cool herps you found or stuff you found online or stuff that you work with, you should totally email me, and we'll talk about it on the shows.
Yes. And if we forget, you can follow up with us live.
Exactly.
There you go.
Like, no pressure, but show my things.
And if you send it while we're on the live, then you can say, hey, I just sent it to you, Kayla.
Exactly. It's okay. I'm down for the pressure. Oh, speaking of which, here's what Tommy sent me. It was which I don't know what kind of mutation that is, but you said that's a Western Diamondback?
It's a Western Diamondback.
Oh, that's so pretty.
It looks like a Yuriko rattlesnake almost. Not exactly, which is a South American rattlesnake. But yeah, I guess that this is the second one to be found in that area over like 3 years.
And which state was it in?
Arizona.
Okay.
Oh, cool.
So it can't go anywhere, and it can't be kept because of their laws.
Yeah.
You can't buy it. I'm sorry?
Was it pretty far south in Arizona?
I'm not 100% sure, but not far south enough that anybody was like, oh, it's a hybridization. Like, because the Yuriko is like almost like Central America.
Yeah.
It would have had to have expanded its range by like thousands of miles.
Then that's unlikely.
Yeah.
Too cool. Yeah, that's gorgeous.
Yeah.
Like that's both of course a color mutation, but that's a pattern mutation as well, isn't it?
It's definitely a pattern mutation also, yeah, for sure. I mean, it's really odd looking.
Yeah.
But yeah, I guess they did some blood and stuff like that on it, and they're gonna check it against other populations in the area, I guess. I don't know, but
yeah, fair enough.
That was really cool. Yeah.
Well,
so cool. Oh, that's awesome.
Successfully done AI with snakes, and if it's a male, you could, even if you can't keep that animal, you could get a sperm sample and try some AI with it.
Oh yeah.
Got to talk to the right people and make it all so it's legal and good, but no reason why you can't squeeze a little goo and spread it all.
Squeeze a little goo. Squeeze a little goo and then spread it all around. Did that just happen on this show?
To put it in professional terms.
How did I
put him in the tube of shame, and then you can just pop out a sperm plugs.
Yeah, no worries.
Yeah, conversations as a grad student, I worked at reproductive biology things, and there are many times we'd be like in the student lounge or whatever, having conversations, or on campus, the food hall or whatever, talking about things and getting weird looks from people next to us. So I understand sometimes, and that's the one time I had someone pass out in my room. It was because I was showing how to get a sperm sample. So that kid hit the dirt.
Yeah, I looked into that because like once like we started having like shed testing and other things like that, like scientific things being attached to like just breeding ball pythons. So it's like AI's next. I gotta read up on it. I need to find out what's happening here.
Beth Roberts at the Memphis Zoo. I've run some stuff for her during COVID, and actually interviewed. I didn't meet her when I interviewed at the Memphis Zoo, but I did interview right when I graduated. Did actually offered me a job to be able to do a postdoc on reproductive work there at the Memphis Zoo, but I ended up going a different way. But yeah, she's done quite a bit of AI stuff. I think mostly with pine snakes, but a lot of that would probably transfer to
I'm sure
pit vipers. So
yeah, for sure.
Yeah, that's cool though.
Let's see. Example.
Sweet Slithering Delights was asking, would you be interested in getting sheds from animals that are from other countries? Like even if the animal's genes are already a test? I guess different lineages.
I don't know that we have a need for that right now, but I would highly encourage anyone, if you have animals, especially for where you are that are, fairly rare, the lineage or what's going on, save sheds, have like a little filing cabinet or even just a little binder thing with, folder things in there, and just take a shed, put it in an envelope, let it dry out, and just file it away. And that way, if that's helpful in the future, that would be great to have. And we do have people doing work similar to what we're doing in other countries. So if there are CITES problems, maybe at some point you just send it to, the more local shed developer, kind of like what we're doing here. With ball pythons, it's kind of a pain because it depends on what country you're in. Some are a lot easier and cheaper than others, but you usually have to fill out paperwork and stuff. And so yeah, it's kind of a pain. But certainly if you have something new and you got a fair amount of it, then we would love to get that into the hands of someone, either us or someone like us, that's going to be doing that development. That's what we're all about.
Yeah. Mike Calderon said that the latest episode we did, he got a shed test of a ball he got from Bob Vu that tested negative for lesser. Yeah.
So you already sent it in. So if you let Sean know your number, your shed ID, whatever it is, you send it in, your order information, send that to Sean, then we can pull it and test it for this new test and kind of verify that for you. But yeah, that's very likely. If it was lesser looking and you got it from Bob, there's a good chance it's this other mutation, which, like we said earlier, may be what the original butter was. But yeah, then you could know for sure. And whenever we get to the very bottom of that story, you'll know what your animal, what the history for your animal is.
Oh, it was a long time ago.
2022.
Ooh.
Still let us know if it was only set in. I didn't even know we had a lesser test in 2022.
It might have been like just when it came out.
Not in 2022, I don't think.
Oh.
It would have been 2023 by the time we had a lesser test, but maybe you sent it in 2022 and then later we ran it for lesser. But either way, if you can find that information, send it. If not, yeah, 2023, that could be possible. But you could also send in that shed again with that original order information to say, hey, can you run this for the new blue-eyed Lucy morph, just so I can, validate it, that is what it is, and we'll happily do that. Right now, soon it will be available as a paid test, and then I have to go through that way. But for the next little bit, we're still trying to figure out exactly where it came from. So that would be kind of nice, just to have a few more verifications from people that think that they have that new mutation. It will be good to see that. I think we were up to 6 or 7 customers that have animals that test for this new test. So
cool. And Sam Saresi mentioned they got their full panel test back. Banana, Black Pastel, Barnhart, Black Pastel, Banana, Leo, Clown came back for a negative for everything else. Barnhart morph was just that good.
Yeah, it can amp stuff up better or at least different than just a normal Black Pastel for sure.
Yeah,
that's very possible. So would shed testing be able to tell us locality of a species or percentage of a locality, like 23andMe? So the way you get to that point of being able to do that is by having a big
genomic database.
Yeah.
So
we would need to build a big database, and we'd need to have examples of animals we know for sure which species or subspecies or locality or whatever it is you're testing. The first species we're working on this with is a what are they called? Panther chameleons. So they did a study where they their whole range, and they took, I think it was like 320 samples. So they took a DNA sample and they took a picture of that animal. So they know what they look like and they know where they came from. And they did sequencing on several genes and looked, and there's actually pretty good separation to be able to tell which locality they came from. So that's probably the first locality test. All the panther chameleon people know we've been trying to get it to the very end. Same with the dry marcon, the Eastern indigo test for identity, be able to have the most genetic diversity when you're choosing which ones to breed together. Those two tests have been very difficult to get all the way to the end, but we're getting close.
Yeah. Would that be helpful also with the people that are doing like super dwarf retics and things like that are doing percentage based like pairings and all that?
Oh yeah.
Yeah.
Because they could also, and then they could also, I guess, get away from like the possibility of somebody saying like, oh, it's this percent this and this percent that, but it's actually not. And then they have a 20-foot snake in their house.
Yeah.
Yeah.
What I proposed to them, I talked to Garrett Hartle specifically about this and some other people in the retic world. What I proposed we do is we sequence, there wouldn't be a ton of them, but there'd be enough, I think, if we did, 50 or 100 or something, where what the end size is going to be for that animal. So an animal people have had either at zoos or people that have been working with retics for a long time. We could essentially run, 25 to 50 that all got massive, and then 25 to 50 that we know stayed small. And then we could have genetic markers that we could test for. And then the kind of 23andMe equivalent could be, okay, this one has a 10% chance of staying under 10 feet or under 12 feet, whatever the line is we draw.
Right.
Or has a 90% chance of staying below. Then it wouldn't really be a locality test, but it would be like a very good measure of, hey, if I buy this animal, what's the chance it's going to stay small? That would be very doable, I think, but also takes time and money, and we haven't really gotten the samples in for that yet. So we'll see if people do it. We could do it for localities too. We just would need 5 to 10 from each locality that you want to be able to test and tell apart. And you have to be able to trust and know. We can't say, oh, well, the exporter said it's this locality. Like that doesn't cut it. Because we know that it doesn't matter what reptile you're talking about, probably other species too, but the exporters are going to just, do whatever they need to do to get stuff sold, not necessarily care whether every animal actually came from that specific location, or if someone brought some from somewhere else and lumped them all together, like they're not going to know.
Oh yeah. Yeah, especially if you're captive hatched. So if they just throw that stuff all together and then sell it off.
There we go. To unlock morphs who asked about emails, I put my email up there for people who want to send stuff for us to talk about on live streams, either today or future live streams. And if you have testing questions or want to get in touch with Sean, you can email him at RareGen Inc.
Yes.
Let's see. Yeah. Making custom snake bag. That's fun.
Right on. We keep clicking at the same time.
Yeah.
Oh, there we go. That's the one I was trying to get. So you got the Genetics 101 has a female that laid 17 eggs. You want to know what locality she is? Yeah, that'd be good to know. And then that's kind of the same thing. We could take information with ball python specifically. We've sequenced so many. We just would need people to give us the information, be like, hey, this female consistently lays over 12 or over 10 or whatever we want to draw the line at. But we could come up with markers to be able to tell, okay, this female's from a line that has a 80% or 90% chance of, being able to lay clutches over 10 or whatever. In the beef world, they're called estimated progeny differences in beef and in dairy cattle. And so the AI is used so much with them. You can have a herd of all cows, whether it's for dairy or for beef. You can just have the females yourself as a breeder and, selling to market either the beef or the milk. And then you can order a straw and you can pay for sperm from this bull that's in another country that's got really, really high genetic merit is very likely to have really good marbling or have, really good milk production. So you can order that in and AI your females from a male that will produce offspring that will perform above average. And all those things are calculated. Like they know they can improve the genetics for time until they're ready to go to market, or how much milk each female offspring will produce, like all that stuff. They have markers for that and they can test for it. And you can have a percent. You can know, okay, if I spend this much more on sperm from this bull, that's going to improve my milk production by this percent or whatever, all that stuff, because that's being used to feed people, and there's millions of breeders that do that, and that's how they make their livelihood. We don't have that much being produced of snakes, so we can't get to that level yet, but we can start making strides that way.
Keep your weird spreadsheets, people. They're awesome.
Yes.
I've got my weird spreadsheets for all my snakes. I'm not as good as updating them for feeding as I was, but things like clutches and sizes, etc. I keep all of that.
Yep, it's good to have more info. Better. And we are getting down to where we're probably only going to be on for like 5 more minutes. So give you your 5-minute warning. Get any questions in. Try to get
Angie Stokely sent us this picture. This is her champagne non-pied animal. And she's pretty.
Yeah, definitely would not surprise me if there's something else there.
Yeah, I wouldn't say GHI though. It does look like something in the 8-ball complex to me though.
Yeah. And there's definitely at least one line of black pastel that nobody can test for. So it could be that, third line. It's not the black pastel we can test for, or Barnhart. It could be that other one that no one has a test for yet. And we're working on it. But that wouldn't surprise me if it was that.
Yeah.
It seems like that's popping up more and more now that it's like, it's a known issue. Like it seems like it's like happening more.
Yeah, especially in Canada, the vast majority, when we, sent that black pastel test information to them, like the only one they got a positive test for was an animal somebody imported from Justin Kobelka. Like everything was negative for that test. And so they have in Canada, almost all of theirs are that line of black pastel that we can't test for. Thankfully, it's more rare in the US, but it's very common in Canada.
Dragon Ruler was also asking, reposting this, have you heard the idea that animals with white on the face have eye issues due to genes coding for the basement of their eye and color being the same or nearby? Do you have any thoughts on it?
It certainly makes logical sense that could be a good correlation.
Yeah.
I don't know. Obviously, it takes a fair amount of money to do studies like that. It would be awesome to see somebody do that with ball pythons. There'd be some really good material they could get and do that kind of a project, but it's expensive, and you got to find, a professor and or grad student that's willing to put in the work to figure that out for sure. But like from a logical standpoint, it makes sense. If the color is changed, then that means the migration of melanin to that place was affected. And we know, like from spiders, that's why they have the funky color, is because the neural crest cells, when they're moving, they're not only moving melanocytes or the melanin stuff, they're also moving the neurological stuff. So you have a weird dispersion of melanin, but you also have a weird dispersion of your neurons, your neuronal pathways and stuff like that. And that's why you get neuro problems. So yeah, it definitely makes good sense that could be the case, but it would be expensive to prove it.
Yeah. Let's see, correlation with pied, bell complex, stuff like palmetto and corns.
Yeah.
Depending on if they have similar mechanisms for what creates that white or lighter coloration.
Yeah, I haven't seen problems with eyes and pieds unless it's like a lesser pied.
Yeah. Me either. And really, the only thing I've seen personally with Blue-eyed Lucy stuff is the eyes being a little smaller, but they seem to
Yeah.
function fine. It hasn't
Yeah.
seemed to affect the animal. It just is a little smaller.
Squunchy eyes.
He got little eyes.
Little peepers. In the world of rats, it's actually considered one of the reasons that an animal could have heterochromia. Interesting. I wonder why it would only affect one eye, though.
I don't know. Maybe it does affect both eyes. I don't know.
That's with albino rats having trouble seeing their heads sway back and forth. I mean, I guess that would just be like the fact that there's less melanin to kind of absorb that extra light they're seeing. So it's kind of a.
That's outside of my understanding.
Yeah. I always thought it was just like there's more light affecting their eyes, or rather, they have less natural sunglasses, I guess. Like the melanin helps with that, I guess. I don't know.
I know the sun burns my eyes out, and I have light blue eyes, and like looking outside without sunglasses makes me want to die.
Oh yeah, walking outside, I get flashbanged by nature. Yeah,
exactly. I'm like, God.
It's like, well, guess I'll die.
Let's see. Any updates on Nyala and mahogany? Not yet. Hopefully very soon. We don't have a lot of Nyala, so it will, if it is in the same area or identical, then we might pick it up. But if it's somewhere different, then we'll definitely need more Nyala sheds. I'm pretty sure we have enough mahogany and super mahogany. We should be able to nail that down with what we have. But we could definitely use more Nyala. I'm hoping that it'll be near enough to something that we can get it or find out that it's the same as something else, but don't know yet. Still working on it.
Cool. Oh, before we close for today, Tommy, you did a thing. Morph map.
I do a thing sometimes.
You do a
thing? Every once in a while. Yeah, morph map. We did LC Black Magic, what, 2 days ago? No? Yeah.
I think so. Yeah.
No, Sunday.
Sunday?
Oh my God, it's Thursday. Yeah, we do morph map every other Sunday at 8 p.m. We did LC Black Magic, and that was a really cool show, and it actually ended up providing more questions than I thought that we had answers, which was really interesting. And even after the show, some people contacted me about the findings that they've had with the gene, or some things that have been happening overseas with it, with testing and whatnot. So it's a really interesting gene. There's like a lot of really cool stuff going on with it. I'm thinking it's in the spider complex. KG thinks it's in the 8-ball complex. So we shall see. We don't know yet, but I'm putting it out there now.
Ooh, exciting.
Yeah, super fun.
Yeah, you guys should go check that out. After this.
Catch the replay.
That's right.
Yeah, exactly. Yeah. All right. So
know what your next one will be on yet?
Yeah, we not this Sunday, but the following Sunday we're gonna be doing Ultramel.
Oh, nice.
Ultramel, yep.
Ooh,
yeah, I'm a monarch girl, but I still like Ultramel. I can do it. So
good.
Awesome.
Ooh. I can hear your laptop better when you're typing.
Aggressive typing.
Oh yeah, I have a mechanical keyboard, so even when I try to type quietly, they're like, "Ugh, it's loud."
I was just saying thanks for watching to all the people on Instagram. Once again,
gotcha.
We will also say after this, when they ask for input, we'll also say once again, like we do every week, please integrate IG comments into everything else. All the other comments are together in one spot, but IG is separate. So I'm the only one that can see them while we're going, and I try to integrate it as much as I can.
Yeah.
But hopefully they'll add it so it's all in one place.
That would be nice. I'm sorry you're the redheaded stepchildren Instagram. You're not to me, and we love you.
That's funny.
All right.
All right, guys. Thank you.
Thanks, y'all.
Thanks for watching.
See you next week.
Bye.